Perfect. All right, rock and roll. Okay, welcome to our our next tutorial here. We are diving into phytoplankton community composition, or PCC, we'll often call it, using our satellite data. Hyperspectral data sets are pretty exciting here because they let us move beyond just the bulk chlorophyll and actually start teasing apart different contributors to the phytoplankton community. So you heard in Sasha's lecture that PACE is soon going to be releasing, into the near-ish future, a couple of new phytoplankton community composition algorithms. And we have, those Jupyter notebooks and Python workflows associated with those, so you can start playing with those. This is really cool stuff. I definitely encourage you all to dive into those. But I know you are consuming a considerable amount of water this week, so we're gonna stick with what's currently being distributed, the MOANA algorithm, or the multi ordination analysis algorithm. So this algorithm gives us near surface concentrations of 3 pico phytoplankton groups: Prochlorococcus, Synechococcus, and then autotrophic pico eukaryotes. And this is all based on, this whole algorithm was built up on empirical relationships that were measured between cell counts that were taken off the ship and in situ hyperspectral reflectances and sea surface temperature as well. These are tiny little guys. They're less than 2 microns, but they're doing a lot of heavy lifting. They're I don't know, 50. I want to say 50 to 90% of the primary production in the open ocean, and about 30% of carbon export globally. So not bad for something you can barely see. But also shows some pretty interesting patterns geographically. Prochlorococcus tends to dominate in kind of warm, low nutrient waters, while Synechococcus tends to pop up along, fronts where communities are shifting towards, bigger eukaryotic cells. And mapping these distributions is really helpful because it can help us spot trophic boundaries, essentially, get a sense of productivity and understand about how the food web is structured. For now, MOANA is only available for the Atlantic, but it's a really handy way to start exploring the pico phytoplankton dynamics here. For all you coastal folks out there, once we've worked with MOANA, we'll take things a step further and look at phytoplankton size classes as well. So this extends beyond the Atlantic and can be implemented anywhere on the globe. Using the standard PACE chlorophyll A product, we can estimate relative contributions of pico, nano, and microphytoplankton. So pico is less than 2 microns, so that all the critters we're talking about in MOANA. Nano, it ranges from 2 to 20 micron, and then microphytoplankton are above 20 microns. So this is not an official PACE product. We're building it ourselves, but it's pretty easy, and it's a great way to think about functional and trophic differences, and it also very closely aligns with how phytoplankton are treated in ecosystem models, where size is an important component here. So long story short, microphytoplankton are basically the fast track to higher trophic levels, while pico phytoplankton tend to dominate offshore microbial loop type of environments. The approach complements what MOANA provides, and it primes you for exploring the more advanced community composition algorithms that are going to be released in the future. So, all right, so we're going to start with MOANA. It gives us detail about the community, dwelling within that teeny tiny pico phytoplankton class. And then we'll layer on the size class perspective that kind of broadens how we think about our phytoplankton community composition. But both are meant to kind of help you connect ocean color to ecology, which is ultimately what we're after here. All right, so you've seen this a few times now. Grabbing our data, we're going to grab an 8-day composite of MOANA and check the file name here. You notice that we're dealing with a level 4 product. So we've taken level 3 data and then made an advanced product with multiple inputs. It takes in SST as well. So we're effectively identifying it as a model if it's a level 4 product. So it's one step removed from a product derived solely from optical imagery. But if we open up the file, we see see 3 separate rasters here. All right. Our Prococcus, Synechococcus, and Pico eukaryotes. Let's see. Load up a few of the libraries here. And we're going to define this little function here too. This just helps us keep our memory at bay by coarsening the resolution. We're going to be looking at like ocean scale maps for this, so we're not going to lose anything vital by doing this. And paste data is also distributed at coarser resolution at 0.1 degree, which is roughly 11 kilometers. We could also grab that instead, but we really wanted to show this little function here just because it kind of demonstrates how you can use Xarray to basically just immediately subsample your data on the fly. And you could just define a program in your workflow here. We're just defining this little function, and we're going to say return an array that's every other pixel in the total image. Essentially, what we're telling you to do. So, all right, so let's make some maps. So we have 3 phytoplankton groups in MOANA. So we're going to create side-by-side plots to compare their distributions. For each group, we'll coarsen the data a little bit to make plotting a little faster and smoother, and then we'll plot it with a color map that's intuitive that we're defining here. Blue for Prochlorococcus and red for Synechococcus, which they can be pink little cells in real life, and then greens for our Pico eukaryotes. And we just define those specific color maps for each. All right, and then we plot this up and get our grid lines and coastlines and borders and whatnot in there, so we can kind of get a sense of where we are. So the result, you get these 3 maps here. We have these 3 phytoplankton groups, and basically we're going to create we're going to kind of look into these a little deeper, but these give you kind of a quick visual sense of where each group tends to dominate. So looking at the maps, you can see that Prochlorococcus tends to really dominate in the offshore water as well. Synechococcus and Pico eukaryotes tend to prefer higher latitudes. Pico eukaryotes also tend to hang out a little more on the coastal margins, at least as much as free floating cells are able to hang out. All right, let's get them a little more quantitative here. First, we're going to clean up some of the memory. So we can, I did forget to mention at the end, this does use the full 2 gigabytes. You will be good on 2 gigabytes, but if you had any memory sort of loaded beforehand, you might want to clear that and rerun and catch up here. All right, so we're going to summarize how each group changes with latitude by using that slice function again. I love this slice function in Python. I'm very appreciative of this, moving from other programming languages. So we're basically going to take horizontal lines of the data and then calculate the median across each of those slices. So conceptually, we're kind of imagine just drawing a line at 50 degrees north. We're going to grab all the values along that line, take the median, then move down to 49.98 and repeat all the way down to 50 degrees south. So doing this for each phytoplankton class basically just gives us a clean line plot that kind of highlights broad, latitudinal patterns, and we'll use that. Like I said, a really cool slice function I like. You may have noticed in this up here as well in the scale, Prochlorococcus, the scale of the cell numbers here, cells per mil, is like an order of magnitude larger than the other 2. So since they're kind of abundant, we're going to put them on we put them on the same. We don't want to put them on the same scale, otherwise it would kind of just dwarf these other 2. So we created a second axis here as part of that when we create the subplots here. So I do want to note that it doesn't mean that they represent more biomass overall, just because there's more cells. Prochlorococcus cells are teeny tiny cyanobacteria. So while Synechococcus and Pico eukaryotes are larger, fewer cells can actually end up contributing more to the total biomass. So Just keep that in mind when you're looking at these relative numbers here. All right. Let's look at things through a time lens here. I really enjoy using this particular feature. So for this exercise, we're going to grab a year of pace data and see how these 3 phytoplankton groups evolve over time. And we're going to use that slice function again, but right now we're just going to extract a little 3x3 degree box in the southern Atlantic. You can pick a smaller area or a different location if you like, but the idea is the same. We're going to take the average of the data, within that box along each time step, and then we'll plot all 3 groups across the year, so you can kind of watch the cycles of growth and decline. This gives some really useful context because not all chlorophyll A is equivalent in this context. A prochlorococcus bloom, for example, represents a very different amount of trophic energy than a bloom of another phytoplankton, and it why you may be seeing that could be a result of oceanographic processes that are happening in the background. So yeah, quick on the code here. Yeah, let's here we defined our little 3x3. Most of this is just creating the plot here. We got a little 3x3 box here, just showing where we're going to extract our data from. Okay, so the code here is there's a really handy tool for dealing with time series. So we're going to define our temporal span here from 2024 to 2025, full year's worth of data, and see how many files we get back. So we get the full 46 here. We're going to use this open MF data set for convenience to load all of these files at once, and it does eat a little more memory than than we might be able to work around in another way. But so for longer time series, if you're looking at like a decade of data, you might consider looping over files and computing median sort of incrementally, so you're only loading 1 file at a time. But that would then take some time. So it's kind of a balance between memory management here. But for a year's worth of data at weekly scales, 46, it's not too big of a lift here. So all right, so we defined our timestamp. Funny little quirk to point out here: the NetCDFs don't have a time dimension associated with them. We could extract this from the file name, but it's kind of hacky, so we'll pull it from the metadata instead. It's a little more elegant sort of solution. So each of these results objects has a temporal extent entry that tells us the start and end of the time period that's covered by that file. So here we're just going to take the beginning of each 8-day window and then use it as the time coordinate. So the list comprehension goes through each file's metadata, extracts the start time, and then trims it just to the year, month, date portion, and then it converts it into a NumPy date time 64 object. So it just gives us a nice clean usable time axis that we are then able to attach to the data set. It's a really helpful portion of this. So it's kind of helping it make as useful as a lot of the other functions we've been using for the satellite data. So yeah, we're able to kind of concatenate the data set and then assign that time dimension that we just defined. So we're stacking it essentially. I will pause here too and note that for ecological analysis, I mentioned this a little bit yesterday, but dealing with level 3 files definitely simplifies things in terms of looking at long-term trends. Each file, is on the same exact rectangular grid, and it makes the files seamlessly stackable and allows them to be manipulated simultaneously. If we're looking at individual level 2 data, it's swath data, so they're not directly stackable you can't really stack them on top of each other unless we sort of project them to a common grid first. It is possible. I know Ian Carroll has some tips for doing this that I can pass on directly with level 2 data, but this is kind of the value of the level 3 files. It's really easy to communicate across images because they're on this common grid, and because of that, we were able to just significantly cut down on our section that we're looking at, and that also helps us save memory. We just extracted this little portion. All the files are stacked, and we were able to just pull a whole grid of those tiny that 3x3 extraction here. Okay, a lot of this. Let's see. So we're taking out that. We have the data set loaded. We're slicing it out here, and yeah, now each of these, you just see that we've taken the median along the lat long dimension. So we're just of that entire box. We're just kind of trying to get one value, one representative value. We could take the mean here as well. It might run slightly quicker. Since we're dealing with cell counts and things might kind of span orders of magnitude, median might be a little more stable. All right. One thing I wanted to mention about that, when we take that slice as well, we're not having to loop through every single time point to do this. With our previous extraction data, we're kind of going through. We had these individual defined points here. It's just we stacked everything by time, and we're slicing right through it, and it happens all at once. So no loops. It just happens sort of all at the same time here. Most of all, this is mostly just plot formatting. You can see but we're going to plot the time series of each phytoplankton type here using the same color scaling we've been using. We don't have to redefine this here specifically since we did it above, but it's nice to keep these cells kind of internally consistent, so you don't have to struggle trying to find what parts of the previous cells you might need to import to make this run. So again, we're setting a different axis for our Prochlorococcus critters, and then we get this simple time series plot over that defined region for these 3 different phytoplankton types over the course of a year. So you can really get a sense of seasonality here and see that the Prochlorococcus tend to dominate during the boreal winter, which is the southern hemisphere summer, because these tiny cyanobacteria thrive in warm, stratified, nutrient poor waters. And then as the year progresses, nutrient inputs from physical mixing and especially upwelling in this region, favor larger picophytoplankton and picoeukaryotes, which can take advantage of the higher nutrient availability. So the seasonal shift reflects a combination of temperature and light and nutrient dynamics, with smaller, fast-growing cells dominating early in the stratified conditions, and then larger cells taking over when the system becomes more nutrient rich. So clean up some memory here. I do want to mention before moving on here, this little snippet of code here is easily adaptable. We did it for one of the West projects yesterday. Replace your product here if you want, chlorophyll L3M _CHL. You replace your time frame. I can't do that right now, can I? Your resolution that you want, 8-day or day, and then you might have to rename a few things below. This would all remain the same here, but as you might have to rename this and get rid of your extra data sets here. But it translates pretty seamlessly with a little adaptation. You can kind of plug and play and make a time series of whatever region you want to look at over whatever time frame. So I think it's pretty useful. All right. So we played with the MOANA functional type approach in the Atlantic. So I want to try a different angle. partitioning phytoplankton by size class. So we're going to use a method by Bob Bruin in his 2011 paper, and this is sort of several iterations of this from earlier, and other folks have kind of adapted this as well. What we're going to do is estimate the relative contributions of pico, nano, and micro phytoplankton from just standard satellite chlorophyll products. The important aspect here is that this works beyond just the Atlantic if you're working elsewhere. So there are a few caveats, though. These are kind of first order global estimates, so that we're going to use some coefficients that were tuned across a really wide range of conditions, but adding regional adjustments, or in other papers they've added environmental variables like SST, has been shown to improve the accuracy for specific areas and regions. You also notice that we're going to be using chlorophyll as an input, meaning we're not actually using hyperspectral data for this. You got me there. The method is simpler than hyperspectral approaches. MOANA does use the hyperspectral reflectance, but it's a very practical way to kind of visualize size class distributions, explore ecological patterns, and really just get a sense of variability and trophic energy potential. As Sasha showed us in the lecture, there's ways of directly exploiting that hyperspectral information to look at different pigments and phytoplankton functional types. For now, since these aren't being distributed by NASA, we want to show you a way to partition the phytoplankton community with just a few lines of code. So in the next steps, we'll compute absolute and fractional chlorophyll contributions for each of these size classes, and make some visualizations that show both the total biomass and then the relative community structure. The idea is to kind of get everyone used to interacting with these sort of multi-dimensional data sets and dealing with not just one type of chlorophyll, right, and not just one type of kind of preparing us for dealing with not just one type of pigment, not just one type of phytoplankton in the future. So all right, let's go ahead and just grab a chlorophyll. 0.1 degree. Quick nuts and bolts here. The next 30, 40 seconds are not too math heavy here, but it's not too scary. Basically, Bob Bruin's size class model uses a couple of just really simple exponential equations to partition total chlorophyll into different phytoplankton size classes. So basically, first, what we're going to do is we compute. There's these constants that are given. These are the constants that might change from region to region, but everything else stays the same. So you can kind of see what those are defined as here. So first, we estimate the combined pico and nano phytoplankton. So this is everything, all the phytoplankton critters that are less than 20 micron. Then we estimate just the pico phytoplankton alone, which is everything below 2 micron. So when we subtract the two, the total, the basically 20 and under from the 2 and under, we get that section in between the 2 to 20 range, which is our nano phytoplankton. Then we take the total chlorophyll and subtract that total amount that was under 20 micron, and then we assume that what was left over is essentially all micro phytoplankton. So once we have these absolute concentrations for each class here, which is what this gives. So CP is chlorophyll for the pico, chlorophyll for nano, and chlorophyll for micro here is what we've put. We can wrap this all up in a data set just to make it a little easier to kind of deal with in terms of interacting with X-array. Now we can attach the coordinates from chlorophyll to it. So now the lat long and all that sort of data is attached to it without us having to manually go through and define that. So it makes things a little easier to deal with. We could also, once we have these absolute concentrations, then we could calculate their fractions of total chlorophyll. It's all we're doing is taking a fraction of each fractional um, chlorophyll and dividing it by the total amount of chlorophyll. It just gives us that fraction within each class. I mentioned, there's potentially some regional specificity to it. But as I mentioned, this logic all here just stays exactly the same. It's only these constants that change depending on local phytoplankton communities. These were tuned globally, so we can, but we can just kind of plug in the numbers and get going. But know that if you're focused on a specific region, it might not be. It's more meant for sort of global oceans rather than maybe a specific area. If you do have enough pigment data of your own, I think they did this with HPLC data, kind of similar to what Sasha was saying earlier. You could tune these for your region and probably get more accurate results. Bottom line is somebody did a lot of the heavy lifting and math already. We just get to use and explore some of these size class patterns and make some cool visualizations. So let's make some maps here. We're going to show a little love to the West Coast here and slice some areas showing us some of the West Coast. And then we're going to plot our 3 size class chlorophyll concentrations plus the 3 corresponding fractions of total chlorophyll. And we're just going to compute those directly from the from this data set that we already pulled, just to kind of keep things. We already have everything in the chlorophyll coordinate space, lat long chlorophyll is. So if we take the ratios here, it just kind of simplifies things. We could treat this like a standard satellite data product and don't have to add things separately. So that's part of the logic behind doing that here instead of taking the fractions at this point. All right. So let's run this. So this 2x3 plot here is kind of just a really quick visual summary. So that the top row is showing our absolute chlorophyll for each size class. So our picophytoplankton chlorophyll, nano chlorophyll, and our micro phytoplankton chlorophyll. And the bottom row is just showing the fractional contributions to total chlorophyll. So these would, if you added all the pixels together, they would add up to 1. And seeing both together kind of helps us understand not just how much biomass is present, but also how it's distributed across the pico, nano, and micro phytoplankton. So that combination of absolute and relative information can be really helpful for interpreting ecological structure and potential trophic pathways in your region. And yeah, set the magic word here, trophic pathways. So let's talk food webs real quick in my little TED talk. So every time, it may be preaching to the choir for a lot of people here, but every time something's consumed, the consumer gets about 10% of that net energy transferred, roughly. So about 10% of the energy from phytoplankton gets transferred to smaller microzooplankton that eat them. And then when larger zooplankton eat those, they only get 10% of that energy, and so forth as energy moves through the food web. In some cases, large phytoplankton can be grazed directly by mesozooplankton, which are the larger class of zooplankton. And this can represent a much more efficient transfer of energy up the trophic food web relative to a more classical steady state system where our microzooplankton or our smaller zooplankton kind of have to be consumed as a middleman before the larger zooplankton can get there. In that latter kind of more classical case, that the larger mesozooplankton only get kind of a fraction of this energy transfer. So it's not as efficient as systems where it's possible for larger zooplankton to kind of possibly graze directly on phytoplankton, making a little bit more of a direct pathway to move energy through the system and get to fish quicker, essentially. there's a lot of, it's a super over a simplification, but there's a lot of other factors that can impact this as well. Alaska is a pretty good example of this. Cold waters, right? You think this might slow down the metabolic energy and lead to sort of less productive fisheries in general, but it's quite the boom of fish up there. So some ecological theory would suggest that smaller zooplankton metabolism does indeed slow down. So their baseline energy demands might not be quite as high as they would be in warmer waters further to the south. This can then reduce grazing pressure on the phytoplankton community, allowing it to kind of flourish and be available to potentially larger zooplankton to graze on them directly. Anyways, more export, quicker pathway to get the fish. If anyone is a biologist, ecologist here, I don't need to tell you that that's not for the faint of heart. And this is like a super gross oversimplification in kind of theory space. And there's all sorts of conditions that throw that out the window. But it's just a good reminder that especially in coastal systems, it's not always bottom up processes. We kind of assume that a lot of times in the open ocean. It's not always going to be nutrient availability that's the top regulator of phytoplankton growth. And also, it's not always more or less chlorophyll that impacts how efficiently energy is moving through the ecosystem. It's nuanced, and this kind of helps you at least get a little bit of a window into that. Anyways, thanks for my I think fourth TED Talk of this series so far. But all in all, it's a great way to visualize the community structure across space and kind of get a sense of which size classes dominate in different regions. And from these maps, you can kind of clearly see delineations across oceanographic boundaries. I just think is really, really fun to look at, where your larger cells are dominating the coastal regions, and smaller cells that much more efficiently recycling nutrients are dominating in oligotrophic gyres. So, all right, back to PACE. One of the trickier things about PACE data is that it's multidimensional. So looking at these three separate maps, we can make a single sort of false true color image, which is kind of neat. When you look at a color image on a screen, every pixel you're looking at, if you zoom in close enough, is actually just a mixture of red, green, and blue light, right? That RGB color system. So if you zoom, you can actually see those pixels essentially lighting up in different proportions, creating color, our different perceptions of in our vision. So if we hijack that same idea for science, instead of using red, green, and blue specifically to represent colors, we can actually use it to represent these three different data sets at once. In this case, we can assign red to the microphytoplankton, red kind of being larger, and then green to the nanophytoplankton, and then blue to the picophytoplankton. So the color of each pixel will then determine how each size class contributes to that location. So it'll be that combination of all three. Now, because these fractions all sum up to one, the colors kind of become really, really intuitive. Bluish regions, as we run this here. So we're just running this RGB image. Using image show tends to work pretty well for this if you're working in image space. But so all three of these sum up to one. So the colors are pretty intuitive here. The bluish regions are pico dominated, the reddish regions are micro dominated, and the greenish are that sort of in between nano. And then the blended colors show the mixed communities and the proportion thereof. So in one image, we can see both the dominance and the transitions, exactly the places where the ocean is doing something really interesting, right, along all those margins. So and if we really want to get fancy, we could also build a little legend that kind of translates the RGB colors into actual fractional contributions of pico, nano, and microphytoplankton. This kind of guide can be really helpful for readers who aren't used to interpreting composite color plots. And it's the type of thing where you to generate something like this that you would want to put into a publication or presentation, so it's just a little more clear to folks what they're looking at. Full disclosure, this particular code snippet was like, yeah, pretty much like 99% AI generated. And that's okay. Don't be afraid to use AI for things specifically like this. There is great value in struggling through problems when they teach you something fundamental, I think. But this is essentially this is a fancy color bar, right? It's nothing to do with the underlying science and everything to do with just making your results clearer to others. So I encourage you not to spend your days combing through Stack Overflow threads or plotting tricks that aren't advancing science. Use your time where it matters. The future is already here. We might as well let it help with the mundane parts. So I feel like old man yelling at cloud here, but anyway, so that's the big picture. MOANA is PACE's first functional type phytoplankton product. It gives us these near surface concentrations of Prochlorococcus, Synechococcus, and pico-eukaryotes. Even if you're not directly interested in those specific groups, they're really useful indicators of the underlying oceanography, especially because the algorithm's informed by environmental drivers like sea surface temperature that go into that. For things like migratory species or identifying trophic boundaries, these patterns could really help flag regions that could be very ecologically meaningful. So yeah, we also kind of looked at this from a different angle by estimating the phytoplankton size classes as well. Pico, nano, and micro, just from the standard chlorophyll A product, and just gives a little bit more of a trophic focused view, helping us think about how biomass is partitioned and how efficiently energy might move up the food web. So yeah, using those coefficients, we were able to kind of get these first order estimates of size class chlorophyll and these fractional contributions. Again, it's not perfect. It could be, definitely optimized for regional applications, but it's a really useful way to connect satellite data to ecological interpretation. You're kind of going a step beyond chlorophyll. So even if there is some uncertainty there, I think there's a lot of value in moving in that direction. So taken together, these functional type, MOANA size class approaches that and the stuff that Sasha was mentioning, the SDP and the GPIG algorithms, they're not meant to be competing ideas, right? These are all complementary tools, and each one tells us something slightly differently about phytoplankton distributions, productivity, and how those patterns might ripple through ecosystems. So I'll leave it at that. I hope this was helpful to you.